Thank you for joining us today for ToxTime. My name is Jenny Rodriguez, Account Development Manager with USDTL. Today's presenter is Dr. Joseph Jones, Chief Operating Officer with USDTL. Before we get started, I'll cover a little bit of housekeeping. This presentation is live, so if you have any questions, please submit them to the Q&A section. The Q&A section should automatically appear on the right side of your screen. If you don't see it, please click the three dots at the bottom right of your screen and select Q&A. Without further ado, we welcome Dr. Jones.
Thank you, Jenny, and thank everyone for joining us here today for a discussion about environmental exposure drug testing, specifically ChildGuard hair testing. Let us begin. Did you know that children living with parents who have a substance use disorder are 2 times as likely to be hospitalized for somatic illnesses, injury, or psychiatric disorders? Did you also know that in this country, we have approximately 2.1 million children living in a household where at least one parent has an illicit substance use disorder? So what's the problem with this? These environments are environments that are unsafe for children. If you have primary caregivers who are more engaged in drug-seeking behavior instead of taking care of their primary responsibility, raising the child, that sets up a situation where you can have a higher probability of abuse or neglect. And the abuse could be physical, mental, or even sexual. Doesn't mean that every kid is abused or neglected, but the probabilities are somewhat higher. So what this means when we break it down is that in our country, 1 out of 35 kids are living in a dangerous drug-using environment. What does that mean for you? If we take the statistic and break it down to a local classroom, that's almost 1 child in every classroom that may be living in one of these environments. What does that mean? The elementary school down the street from your house that maybe your child went to, is someone in his or her classroom in one of these environments? Does your child visit their home or spend the night? Does that child come to your house? The average elementary school in this country has between 500 and 600 students. So that means in the elementary school right down the street from your house, there could be up to 14 children in that elementary school suffering in plain sight. So what can we do about it? Well, USDTL's vision is to protect and enrich lives. And we're on a mission to make a difference. And we've been doing this for a while. And we got started in this back in the 90s. Hair testing was just getting started. It was just becoming accepted in a lot of different venues. And we had a child protection services manager in a county and a state here in the Midwest. And they called us. They said, we've got a case here that is being difficult. We knew that the child was in a dangerous environment, but the parents were very slippery. They were unable to get them on a positive urine drug test. They knew how to beat it. They knew how to avoid drag their feet. When a hair test was ordered on them, Dad showed up shaved from head to toe. Mom showed up as a platinum bleached blonde and shaved from the neck down. And so thwarted the efforts at that point. So they called our laboratory and they said, we keep hearing this thing about environmental exposure and that it's an issue with hair testing. In this case, do you think that if we got a sample of the child's hair, do you think we would pick up environmental exposure? And we said, well, of According to the literature, it should. And so we gave it a try. CPS went to the school, got the child, got a hair collection, sent it to the laboratory. We analyzed it, and we reported it back out as positive for methamphetamine. Talking to the CPS manager, they're like, thank you, we can take it from here. So that first test, the very first ChildGuard, we were able to provide concrete evidence, conclusive evidence that this child was in a dangerous methamphetamine environment. And from there, we've grown from that very first specimen up to between 500 and 600 cases a month now. So before we go on, I would appreciate everybody responding to this poll question. I have two questions. And the first one, and it's just yes or no. Have you ever provided drug testing services in a child custody case? Yes or no? And the second question is, have you ever used ChildGuard or worked with someone who has? If you could please answer yes or no, then we can move on. I'll give you just a second. That'll help me calibrate my presentation going forward. Thank you guys very, very much. So in order to really understand how ChildGuard works and environmental exposure testing works, it would pay for us to circle back and talk about how are drugs incorporated into hair. So drugs are incorporated into hair through three main routes. First and foremost is environmental exposure. If you're in a room where drug is being smoked, handled, distributed, excess particulate matter, that drug will migrate to your hair, get on your hair, work its way into the nooks, crannies, and pores, bind to the proteins and pigments, and then it's available to be harvested and analyzed immediately. Route number two, sweat and oil from the scalp. These fluids contain drug and drug metabolite. This is important. Drug and drug metabolite, similar to urine. And as those fluids bathe the hair shaft, they deposit the drug and drug metabolite. Again, it works its way into the nooks, crannies, and pores, binds to the proteins and pigments, and is available to be harvested. We start seeing that maybe 2 or 3 hours following a dose. Starts to show up. And then route number three, which is what we normally think about with hair testing, is the blood, which contains drug and drug metabolite, deposits drug and drug metabolite into the root. And as that portion of hair emerges past the scalp line, which takes about 2 weeks, it can then be harvested and analyzed. So at the end of the day, we've got three extremely different modes of incorporation superimposed on top of each other. So it's a very complex picture. And so right from the beginning, we are not able to backtrack to determine time, dosage, or frequency. There's just too many variables. Now, hair at average grows at a ½ inch per month, especially at the back of the head where we collect these specimens at the laboratory, we trim that hair to 1.5 inches and we discard the excess. So our detection window is limited with up to approximately 3 months of hair growth. Most drugs stick around a little shorter than 3 months, a little longer than 3 months. 3 months tends to be like a good happy medium for detection. And it has become a de facto standard in our industry. Now, the next question that we get is, can you use fingernails? Fingernails are keratinized proteins, very similar to hair. And yes, you can. You can use nails, but it is less sensitive. One of the variables in hair testing is the pigment. And fingernails generally have little to no pigment. It is also a beta pleated sheet anatomy, as opposed to hair, which is an alpha helical. With the With the fingernails, you have this one surface on top for the contamination. With hair, you've got 360 degrees around the shaft of the hair. So for a couple of different reasons, hair seems to be a little better, a more sensitive assay for the detection of environmental exposure. So here comes the big question, right? So we get this result back. Was it environmental exposure or was it ingestion? Now, there's a couple of different strategies that different laboratories use to answer this question. The earliest and one that is still in use is called wash kinetics. And what this means is that you wash the hair a certain number of times with a certain buffer, and it will remove the environmental exposure. There's been enough data in the literature to show that that does not work every time. And for that reason, we do not subscribe to that strategy. The strategy that we prefer to use is called metabolic profiling. Big term, simple concept. If we find the drug itself in your hair, that is evidence, it's conclusive evidence of environmental exposure. However, if we find a unique liver-produced metabolite in your hair, then that is evidence that the drug was inside your body and therefore it had to have been ingested. So that's, to me, seems to be a little simpler way to go. And our laboratory, this is our preferred way to interpret these results. So what does this mean when you get a result? We can look at a few examples here. Let's take the most prevalent one, marijuana. If we find THC, THC is the drug that's in the leaf and the smoke. This is what makes you high. If we find that in the hair, that is evidence of environmental exposure. Doesn't mean you didn't use, we just don't have any evidence of that. However, if we find carboxy-THC or THCA, that's a unique liver-produced metabolite. That means it's been inside the body, so it had to have been ingested. So to review, if we find THC in the hair and that's it, environmental exposure. If we find THCA or carboxy-THC in the hair, that's evidence of ingestion. Let's look at another example. Methamphetamine. Methamphetamine is metabolized to amphetamine. The reverse is not true. So if we find methamphetamine in your hair and that's it, evidence of environmental exposure. If we find methamphetamine and the unique liver-produced metabolite amphetamine, that's evidence of ingestion. You'll also have some cases where people are taking Adderall or Vyvanse, which is pharmaceutical grade amphetamine. Now, they will only have amphetamine in their hair, not methamphetamine. But it can get tricky. If you need help, call the lab. We'll be glad to help. Another example is hydrocodone and norhydrocodone. If we find hydrocodone in your hair, conclusive evidence of environmental exposure. Norhydrocodone is a unique liver-produced metabolite. Same thing with oxycodone. Drug in your hair, environmental exposure. Noroxycodone, unique liver-produced metabolite. Now you might be thinking, how does someone get oxycodone environmental exposure in your hair? Several of our counts are medical professional substance abuse monitoring programs, and we've had occasion where we've had pharmacists who are handling these materials without gloves on, and they get contaminated with the oxycodone on their fingernails, in their hair, but the noroxycodone is absent. However, if they are relapsing or diverting to themselves oxycodone, we've had cases on both sides of that. We've been able to demonstrate environmental exposure and ingestion in different cases. So this is very helpful. And now a drug that has been in the press a lot in the past few years, fentanyl. If we find fentanyl environmental exposure, norfentanyl, a unique liver-produced metabolite, evidence of ingestion. We may see fentanyl as environmental exposure in some cases because if they're smoking cocaine or if they're smoking heroin that's been laced with fentanyl, we may find that in the hair of the child. So this is good information to have. Now, here comes the difficult ones, cocaine to benzoylecgonine. Benzoylecgonine is the primary metabolite found in urine. It's the principal metabolite of cocaine. Unfortunately, not only is it a metabolite, but it's also a natural decomposition product when exposed to moisture at the right pH. And so in this case, benzoylecgonine is not a unique liver-produced metabolite. It can be there from different regions. So we can't use benzoylecgonine to help us interpret cocaine. Same thing with heroin. Heroin on standing with moisture at the right pH will decompose to 6-mem. So these two are complicated, and this can be the subject of an entire presentation. And so we'll hold off and talk about those in more detail at a later date. If you have one of these results, call the lab. We'll be glad to help you work through it. Now, one of the questions that we get sometimes is, do these tests hold up in court? And the answer is yes. And that's because we follow two of the primary constructs in producing a forensically defensible result. Construct number one is chain of custody. Every specimen at USDTL is collected, received, processed, reported, and even to when we destroy it 1 year later, all of that activity is done under a documented chain of custody. And we can reproduce that data and show exactly who had it, where was it at any given point in time. So that's important, chain of custody. Equally important to chain of custody is confirmation testing. All positive results at USDTL are confirmed by an appropriate mass spectrometric method. That is the gold standard for confirming drug results in a forensic environment, and all positive results are confirmed by mass spectrometry. So taking these two constructs in tandem, these are the cornerstones of generating a forensically defensible result, and this happens on every sample at USDTL. These are two questions that are really important to ask once you get into any kind of drug testing situation. Litigation support can be tricky, and so we're here to help. We can assist you with business record affidavits, letters of interpretation, litigation packages, which are certified true copies of all of the raw data and the chain of custody, both internal and external to USDTL. And we have a team of scientists that are there to help you with expert testimony in case we need to either testify via telephone, video, or on site. We're there to help. So what trends are we seeing at USDTL? In our ChildGuard for 2020, last year, these are the positivity rates that we observed in our ChildGuard. Now, keep in mind, this is not a general U.S. population, general demographic. This is a very select, high-risk population that we're testing. Child custody cases where drugs have been implicated, suspected abuse, neglect. So these are not run-of-the-mill general population results. But as you can see, marijuana is by far the most prevalent and up over 70%. We have methamphetamine at almost 64%, cocaine at 7.7%, and opiates at 1.8%. So on the methamphetamine, something interesting is that over the past decade, the DEA has been reporting that they are seizing more and more methamphetamine at the border. And you can see just how much it has gone up over the past decade. Also, they're reporting that the purity of methamphetamine at the border is going up and that the price is going down. So this is not a good thing. During the same period of time, methamphetamine-related admissions into public treatment facilities has been on a steady increase. And Quest Diagnostics reported in their drug testing index, specifically for hair, general workplace, positive methamp has been going up there as well. So this begged us to ask the question. Now, if you're not familiar with United States drug testing laboratories, One of our significant lines of business is what we call newborn toxicology. We test little babies right after they're born, either using their meconium, which is the first little poo a baby takes, or we get a snippet of their umbilical cord, about 6 inches. And we can do a drug test that reflects maternal usage during the, or in utero exposure during the last half of the pregnancy, approximately. And so when we saw these previous slides, we asked the question, would a similar increase be observed in a national data set of umbilical cord tissue results? And this is what we found. We found that we did indeed see a dramatic increase of methamphetamine over the last decade. And we can see at the beginning of the decade, it was plateauing at almost 3%. At the end of the decade, it's plateauing in the mid 5%. And we had this rapid increase between 2014 and 2017. What's going on there? The news was full of the opiate epidemic, not the methamphetamine epidemic. So what was going on here? This data was reported at the annual conference of the College on Problems of Drug Dependency last June. In preparing for this presentation, I was thinking, would similar increases be observed in a national data set of ChildGuard hair analysis? And so we pulled data for the last couple of years, and lo and behold, we did. It's a very similar pattern. We have kind of a plateau at the beginning of the decade at around 50%, and we're at the upper 60s here in the last few years with this rapid increase between 2016 and 2018. The years are a little different, but it's the same general pattern. Don't know what to make of this, but this can't be a good thing. Methamphetamine is not a good thing for the prevalence of it to be increasing in a population. So to me, this dramatically increases the importance of ChildGuard. And so now on to probably the funnest part of a presentation like this is let's take a look at some real case results, and we can interpret the results, and you can see how you can use these results in your jurisdiction. So let's start with our first case. So this was a program ordered abstinence. So this person was either in like a board of nursing program or physician health program, airline pilot program, somewhere where it's safety sensitive, and they've been ordered and they've signed a contract to be abstinent and not to associate with known illicit drug users. So the participant had a head hair that was negative for amphetamine and positive for methamphetamine at 7856 picograms per milligram. So interpretation on this is we've got the drug, no metabolite, environmental exposure. But then the toenail and the urine, and frankly, this was several urines, were all negative. So how could that be? Well, urine it will not pick up environmental exposure. And with the toenail, if you're wearing socks and shoes, it's not exposed to the methane as readily. So this story kind of fits. The donor suggested that the significant other was still using methamphetamine, and they asked them to submit to a drug test, and there it was. So now we could deal with the situation. This donor was not obliging to the contract. They were associating with a known methamphetamine user, and I don't know what happened following that because we're kind of cut loose at that point. But at least the caseworker was able to work with that participant with full knowledge that, you know, if you're associating with a known user, that's like the first step to relapse. So it's just not a good idea. So our second case was a pharmacist. This was the pharmacist case I was telling you about, where the head hair had 782 oxycodone, but no metabolite. The fingernail had 256, but the toenail was negative for both oxycodone and noroxycodone. Again, this kind of fits that pattern we're talking about, where the head hair and the fingernail, especially if they're handling the pills with their bare hands and not wearing gloves, but then the toenail is under wraps under a sock and shoe. And so this kind of fit the story there. Now here's a drug court case, and where the drug of choice was marijuana for this particular participant. So we had a head hair at 421, and he also had a carboxy THC at 0.84. So this is consistent with ingestion. They also had a fingernail that was at 1525 THC with 2.54 for the carboxy. Again, consistent with ingestion. And then we had a toenail at 2502 and the metabolite at 9.86. So all three specimens were consistent with the participant continued use of marijuana. But the interesting point I wanted to show you here is that when we have someone that's using a particular drug, we will typically see, like it, especially for marijuana, we will see where the hair is lower than the fingernail and the fingernail is lower than the toenail. And it has to do with the thickness and that the drug is protected by the thicker nail material. The fingernail is like 4 times thicker than that strand of hair. So it's less opportunity to leach out when you're normal daily hygiene. So I thought this was an interesting case for us to take a look at today. Here we have a DCFS investigation where we have a participant's head hair with 1126 and the metabolite at 0.12. So that's consistent with ingestion. We have a 2-month-old with a head hair at 168 for the drug and 0.08 for the metabolite. So ingestion. And we have a 5-year-old that only has the drug at 423, but negative for the metabolite. So here we've got the participant consistent with ingestion. We've got the 5-year-old consistent with environmental exposure. But then we've got this 2-month-old that looks like it could be ingestion. So there's a couple of things. When we got the little kids, we have to be careful with these. So number one, hair goes back 3 months. And so this could have been exposure in utero. Number two, or ingestion in utero. Number two is that the sweat and the oil from the hands of the mother, with a little infant and you're holding it all the time, the sweat and the oil can contaminate the child's hair. It's potentially there, so we have to be aware of that. Another thing is that the baby could have been breastfeeding. In this case, the baby was not breastfeeding, so that was not the answer. We knew that the child was exposed because of the 5-year-old, and so the ingestion probably was either in utero or from the sweat and the oil from the hand of the mother. And for the last case study here, this was a child custody case. Mom was accused by dad for using methamphetamine. Mom's head hair was at 1276 amphetamine, methamphetamine at 7856. So this is consistent with mom ingesting methamphetamine over the 3 months prior to the collection of the specimen. We had dad with a classic result for environmental exposure with negative for the metabolite and 1256 for the drug. And then we've got 3 children. We've got a 6-month-old that was negative for the metabolite and methamphetamine at 4500. A 3-year-old negative for the metabolite, 850 for the drug, and then a 15-year-old child that was just negative. So we've got a case here where we've got evidence that mom was using, dad was exposed, and 2 of the 3 children were exposed. Well, what about this 3rd child? What's going on there? And so I put this case in because we see this phenomenon quite a bit. And I call it stair-stepping. And so you will get the highest results for the youngest children, and the results will continually decrease as the children are older in the household. And as the child develops, they tend to migrate farther away from the caregiver, average distance away over time. And so we've got here an arm baby that was positive, and then we've got a 3-year-old who's out and about playing and not necessarily in mom's lap arm all the time. And then we got the 15-year-old who, I don't know, maybe was playing Little League baseball or basketball and just wasn't at the house much. You'll see the results drop down like this. And sometimes, like the older children, will be negative just because they're not around that parent that much. So this brings us to our last case study, and it's almost to the end of our presentation here. And before we close out today, I wanted to take this opportunity to invite each of you to join USDTL in our mission to make a difference, on our mission to protect and enrich lives. And so how can you do this? Well, first, you could visit our website, and the link is at the bottom of the slide here. And clients have told us that they have appreciated the amount of information that we've accumulated there over the years with regard to hair testing in general and ChildGuard testing specifically. And so you're free to review that data, pull it down, use it in your jurisdiction how you see fit. If you see any gaps, let us know, and we'll find additional information and post there. We're adding to it all the time. Second, I also hear from clients that, well, I didn't know you could do that. So take the opportunity to either send us an email or call the number listed below and talk to our client services and go over our catalog or request a catalog. Be happy to tell you all the different panels and profiles that we have. Did you know we have a 19 panel in hair? It's new. If you are a client of USDTL and you've never ordered a ChildGuard, call your account manager and they'll be happy to walk you through and talk you through how to set up ChildGuard and how to order it. And last but not least, if you're not a client of USDTL and you would like to use our services, again, send us an email to the email below or give us a call at the 1-800 number below, and we will be happy to have someone walk you through setting up accounts so that you can send samples to USDTL. And so with that, that comes to the end of my presentation, and we can now shoot this back over to Jenny. Jenny, take it away.
Thank you, Dr. Jones, for that valuable presentation. I would like to take some time to present to you guys our moderator, Grace De La Peña, who is our account manager with USDTL.
Thank you, Jenny, and thank you, Dr. Jones, for that valuable presentation. I will give our audience a few moments to submit their questions. As a reminder, if you don't see the Q&A section, please click the 3 dots at the bottom right side of your screen and select Q&A. While we wait, I want to let the audience know we will be sending recording of this event for everyone that registered. For those attending this event live, you will receive an additional email containing a certificate of completion within the next few days. So let's get started with some questions. I do see some coming through. Dr. Jones, first question. Our current lab has always told us that environment exposure could not be tested. What does USDTL do differently from other labs to test environmental exposure?
This goes to the heart of the matter that I mentioned a few minutes ago, in that there are 2 different schools of thought on how to interpret hair results. The other laboratories in our country focus on workplace testing, where they are not concerned with environmental exposure, and in many cases, they want to mitigate that as much as they can. So they will tell you that they wash off all of the environmental exposure, and they will tell you that they will not pick up environmental exposure. I don't buy that. I've seen too much samples back and forth between the laboratories, and the literature does not prove that out. It does work in most cases, in my opinion, but in your specific case, that may not be the deal. Now, in the case at USDTL for ChildGuard, we don't wash it at all. And so we want everything that's on that hair to make it into the test. And so that's why we pick up environmental exposure, and the other labs say that they don't.
Thank you. Next question. Is toenail window of detection the same as fingernails, 90 days?
No, good question. Fingernails are up to 4 to 6 months for drugs of abuse, 3 months for ethyl glucuronide, a metabolite of ethanol. Toenails can go back 10 to 14 months. Some people say even longer. So toenails have one of the longest detection windows of anything that you can test.
Great, thank you. I have another one. I have had a client admit to heroin use, but it does not show up in the hair follicle. What does this happen? Why does this happen?
Okay, so if we could start with like the previous question, if it went to another lab, maybe they just washed it all out, ingestion and environmental exposure. So that's possibility number one. Number two, maybe they didn't use enough. And so single uses do not necessarily show up on a hair test. Maybe it was outside of the detection window, or maybe they cosmetically treated their hair. Bleaching, perming, dyeing, chemical straightening, all of these processes contain varying amounts of oxidizing and or reducing agents. And depending on the brand and how it's used, you can destroy the drug, maybe all of it or just enough to knock it below the cutoff.
Gotcha. What's the difference between using a ChildGuard versus the regular hair test?
For the hair test, we do a wash, and it's mainly for regulatory reasons. The popular vernacular in the hair testing field right now requires that you do a wash before you do any analysis. We would prefer just not to do that and skip it all the way around across the board, but our regulating agencies will not allow us to do that. So for non-ChildGuard, we do a wash in the organic solvent, and we know that washes off a good portion of environmental exposure, but we know it doesn't wash it all off. And so at our laboratory, we report what we find, and then we help you interpret the results afterward. For the ChildGuard, we don't wash it all. We want to optimize the sensitivity for environmental exposure. We skip the wash step, and everything that's in the hair makes it into the test.
Thank you. In the case of substance that does not have a unique liver metabolite, such as cocaine, wouldn't the cutoff level help to determine if the substance was ingested?
Yeah, so there's a lot of different, and the cocaine is one of the more difficult interpretations that you can have. So the mere presence of cocaine in hair and nothing else, environmental exposure. Benzoylecgonine can be present because of natural decomposition. Cocaethylene can be present due to the original drug being contaminated with trace levels of cocaethylene, and small trace levels of norcocaine can be present. So this is a problem. This is a difficult thing. And so at our laboratory, we use the ratio of norcocaine to cocaine at 3%. If it's above 3%, it's highly likely from ingestion. If it's 3% or less or not even there, then that is environmental exposure.
Okay, thank you. Next question, I'm going to interpret this a little bit. Can adults be tested for environmental testing?
Absolutely. One of the cases that I showed as a case study, the mom, the dad, and the 3 kids were all ChildGuard when they came in.
Is there a way to distort, mask, or cover up a hair follicle drug screening?
Yes. Bleaching, perming, dyeing, chemical straightening.
Does excessive swimming affect the hair for testing?
It could. You got chlorinated water, and the detection window for drugs in hair is based on normal daily hygiene. A swimmer that is like, say, like a high school or college swimmer who's swimming 2 times a day, plus taking a couple of showers a day, then it's going to wash out faster than the average person. So yes.
Okay. When it comes to substance, aside from the main drugs that are more commonly known, can drug tests identify the newer combination of drugs such as bath salts and other more newer drugs? If so, how are they identified as compared to the other known drugs through USDTL?
So this is a very, very difficult situation. These novel drugs that have come out, synthetic cannabinoids and bath salts, they change so rapidly that by the time you develop and validate a new assay, it's irrelevant by the time you get it to market. The saving grace for synthetic cannabinoids specifically is that with urine testing, most people who really, really like their marijuana can go 3 to 5 days using synthetic cannabinoids in order to pass a urine test. However, doing without their marijuana for 3 months is a whole lot more difficult. They don't like the synthetic cannabinoids as much as they like their weed. And so what we find is that you just pop them on marijuana, so we still get them positive. There was a paper that came out of Italy that showed that in a large survey of that population. And so the JWH compounds that all hit the news big time 5 or 7 years ago, you don't even see it no more. They've moved on to these other alphabet soups of compounds, and the validating and developing these methods is incredibly expensive. And by the time they get to the market, they're irrelevant, and then nobody wants to test anymore.
Thank you. Next question, can body hair be tested for environmental exposure? If so, what is the detection window?
Yes, and so the detection window is much more varied, but it can be up to a year. So sometimes that's too long. It's probably not going to be a year, but because it is body hair and there is such huge inter-individual variability, we have to assume that it could be up to a year. So you kind of have to understand that to see if it's even worth your while to send that sample in or not.
Thank you. If a child is removed from a parent's home and their hair is washed multiple times, does it affect the results of the ChildGuard?
Yes, it could. The 3 months is due to normal daily hygiene, and the environmental exposure, if you just kind of do the thought experiment, is going to be easier to wash off or wash out than what's been laid down from ingestion. And so we kind of see this once in a while in that we'll get a sample, you know, they catch the parent off guard, they get the sample from the kid, and then they show up a week later and lo and behold, it's negative. And so I'm quite certain the kid spent most of the week in the shower with mom scrubbing him pretty good. So that it's normal daily hygiene is 3 months. And then again, what's normal daily hygiene? How do you describe that?
Okay. I recently had an adult say their hair follicle was meth positive because they used meth for 31 straight years, but quit 6 months. Is this possible?
It would depend on the situation. And I've had a case like this. I had a meth amp user who was, you know, using very, very heavily for a very long period of time, like what you just described. And so this person was successful in the rehab. They came back 3 or 4 months after rehab back to the house where they were living at. Well, that house was so contaminated that it recontaminated his hair. And it took several months for the environment for the drug to sink out of there and then for him to go negative. So it took him 6 or 8 months before he was able to give us a negative specimen. But that was kind of a unique situation. It was very, very heavy use. And we had him wash like his hoodies and his cap, get rid of his caps, upholstery pillows and things like that. And so that kind of helped him get clean. But if they go back to that same environment, you know, the environment can recontaminate you. So you have to be aware of that.
Okay, thank you. Here's another one. If the exposure was on the later end of 3 months, would you expect the result to be lower? For example, THC?
We would expect it to be lower, but here's the $1 million question. How much was it when it started? And that we'll never know. One of the things that we can see, and we should never use hair and nail results. We'll watch them go down. You shouldn't do that because the hair and the nails, they speed up and slow down according to health status, seasonal, diurnal, you know, and so there's lots of reasons why they speed up and slow down. And if they're growing slower, more can accumulate than when they're going faster. in a period of time. And so we have to be aware of that. And we can see that result bounce around somewhat. It's called pulsing. And so especially with fingernails, you can see them take wild swings going up and down.
Thank you. I have here, would you recommend exposure testing to be ordered in addition to standard hair panel for those who are requesting a look back at previous failed tests? Would you recommend a combination, both testing the environmental exposure, the ChildGuard, or the standard hair testing?
I would say just order the ChildGuard, because with the ChildGuard, you're going to get environmental exposure, and you will also get the opportunity for an ingestion interpretation. And since we're not washing anything off, any detection, any use that was like a long time ago, it would improve the odds of it. Doesn't guarantee it to show up, but it will improve the odds that it would show up. So that would be my recommendation if I understand the question correctly.
Okay, yeah. As far as the results, when the results is positive, how do you report out environmental exposure on the report?
Good question. We don't. We report what we find, and the report has only objective toxicology data on it. We do not do any pre-interpreting. We do not do any subjective interpretations. We can do that on a letter of interpretation. But as far as the report itself, it's the objective data, and then we can interpret that after the fact.
Okay, thank you. We have a couple more minutes to answer a couple more questions. So if you have additional questions, feel free to submit those to us. I do have another one that just came through. If someone had a positive ETG, one set of 3 months, the next 3 months is still positive. I believe that's, I don't know if you can, the person is not, sorry, it's going through repeatedly. So they're asking, as far as the positivity of the ETG, how does it go every 3 months? If the individual is thinking 3 months and then not thinking, how does that show as far as the results?
So the data that I've seen with ETG in hair is that it doesn't quite survive 3 months in the hair. The data that I've seen. And so and that's from normal daily hygiene. So if someone jacks up their hygiene regimen, they could probably become negative for ETG hair much quicker. So that's number one. Number two, there are, as they call them, hair lotions, and I'm not quite certain what a hair lotion is, but the literature out of Europe says that in Europe, anyway, there are hair lotions that contain ETG. So they're plant extracts that are extracted with ethanol, and it turns out some of those plants have the enzyme that forms ethylglucuronide. So you go to the dispensary in Switzerland and you get your hair lotion, and that stuff has ETG in it. And so you put that on your hair, now you've got that, but then it should wash off a little easier than if it was laid down by sweat, oil, and by blood. So I'm speculating here, but those are a couple of reasons why you could have like a spot positive ETG in the middle of a bunch of negatives. Another reason could be lab error. And so as always, and before we end today, always, the last line of defense for any donor is the retest. If a donor refutes their drug test, it is inappropriate to collect a sample to refute that, because a drug test, even though we've got long windows of detection, they are a snapshot. And so the appropriate way to refute a drug test is to have the offending specimen retested either at the laboratory where it was originally tested, or more appropriately at another laboratory, and let's see what they have to say about it. Invariably, if you give people enough time to study for the test, they can pass it. So don't forget that. I've seen that too many times.
Okay. Next question. Can CBD oil or edible show up in the drug screening?
Yes. So the CBD itself, not so much if it were pure. But here's the problem. there's no such thing as pure CBD products on the market. They are all contaminated to varying degrees with THC. So when we find the THC in the hair, the products are contaminated with THC, so you're just taking THC. It might be a low dose. You have to take a lot of the gummy bears or whatever to get positive, but nonetheless, over the long haul, you're just taking THC along with the CBD. Now, this is a very, very difficult problem to solve, and we've validated a couple of specimen types to address this to help us with the interpretations, but it's a very tricky assay. And now that CBD is becoming more prevalent in our society, one of the things that the DOT did early on last year, and I'm glad they did this, was they just made a blanket statement and said, you know what, these products are contaminated with THC, so the DOT does not accept CBD products as a reasonable explanation for a positive carboxy THC in a federal mandated urine. So I was glad they did that, because then that cut down a lot of the CBC discussion at that time. But over time, it's going to be required of the laboratory to be able to make some sort of a statement about what happened. And so we're not there yet, but the day's coming.
Great, wonderful. We have one more question.
Okay.
Thank you so much, everyone, for the question. Are you aware of any study on the impact on children of environmental exposure that you would recommend for us better understand the impact of the exposed children?
Yes, and there's a couple of papers out there that talk about the negative impact to the children. One of in the very first slide, you can see when you get the copy of our slides when they're made available, that reference at the beginning talks about the hospitalization. The odds of a child in those environments, the hospitalization is higher. There's also some other references and that were part of a newsletter article that we wrote a couple of years ago, where they talk about that children in these environments are 2 times and 4 times more likely to be exposed to abuse and neglect. And so that data is out if we find an illicit drug in their hair. So that one was a little broader, but a little more specific at the same time, where you find illicit drug in the hair, 2 times and 4 times more likely to be exposed to abuse and neglect. We'll make links to those papers available on our website. And so go to our website, and if they're not already there, we'll make sure that they're up top so you can see them easily.
All right, thank you so much, Dr. Jones, for providing everyone valuable information about ChildGuard. If I was not able to read off your question, we will be responding to you directly. I want to mention to the audience that we will be conducting another ToxTime live next quarter. We would love to receive some feedback about this event by completing a survey once the event ends. To keep up with the latest event with USDTL, we encourage you to visit our website and join our mailing list. Also follow us in LinkedIn, Facebook, or Twitter. We hope everyone enjoys today's presentation. Thank you again for joining us, and have a great day.